11 years of cancer research has proven fruitful for scientists at Johns Hopkins who uncovered a new tumor suppressive response that could lead to novel therapies targeting hard-to-treat cancers. The new study, funded in part by the National Cancer Institute, showed that targeting a key process of how cells make proteins can inhibit cancer cells and resolves what makes them so sensitive. The findings, published in June 18, 2025 in Cell Chemical Biology, opens the door to potential new treatments for cancers with common genetic mutations. The researchers found that using a drug to inhibit the enzyme responsible for human RNA transcription called RNA polymerase I, or POL1, triggered a unique stress response that rewires splicing, or the way cancer cells produce forms of proteins, to ultimately suppress tumor growth. Ribosome biogenesis has long been known as a hallmark of cancer, says study leader Marika Laiho, MD, PhD, a Johns Hopkins professor of radiation oncology in the Department of Radiation Oncology and Molecular Radiation Sciences. In 2014, she and her team identified that POL1 is a meaningful therapeutic target in cancers. She began laboratory studies using human cell lines to study a small molecule, BMH21, developed together with Johns Hopkins pharmacology and a molecular science expert, James Barrow, PhD, to inhibit the POL1 enzyme. In the latest study, the team analyzed more than 300 cancer cell lines and found that tumors with certain mutations were especially sensitive to POL1 inhibitors, such as BMH21 and a new drug called BOB42. These alterations are common in cancers with mismatch repair deficiency, MMRD, including colorectal, stomach, and uterine cancers, which results in copying errors going uncorrected when DNA replicates and cells divide, causing high rates of additional mutations and cancer risk. The team tested the new drug in animal models and patient-derived tumors containing the same key genetic markers and recorded a significantly reduced tumor growth by up to 77% in melanoma and colorectal cancers. These findings highlight a promising new path for targeting cancers, especially for patients with mismatched repair-deficient cancers that are resistant to existing therapies, says the study's author Wenjin Fan, PhD, research associate. The study also suggests that changing how cancer cells splice RNA or produce different forms of proteins could affect how the immune system recognizes tumors. Combining immunotherapies with POL1 inhibitors may improve the effectiveness of immunotherapies. This is an entirely new conceptual framework for understanding how RNA synthesis influences cancer cell behavior, said Dr. Laiho, who holds patents in POL1 inhibitors that are managed by the Johns Hopkins University in accordance with its conflict of interest policies. Our study reveals that ribosomal protein RPL22, typically a structural component of the ribosome, plays an unexpected dual role as a critical regulator of RNA splicing. Targeting this pathway can not only suppress tumor growth, but also moderate tumor antigenicity, enhance responsiveness to immunotherapies. If you like more daily doses of positive news, please subscribe to my channel. Please like and leave a comment, as this really helps the channel grow. Links to the article on this news story can be found in the description of this episode. Thank you for watching.